Well, aloha everyone, and welcome to my lecture on metabolism and nutrition. Metabolism is the entirety of all of the chemical reactions that are taking place in the human body at any given moment. And some of these reactions are going to break things down, and some of these reactions are going to build things up. So if you can remember that anabolic steroids pump you up like Arnold, anything that's going to be putting things together or building are going to be anabolic or synthetic reactions. And reactions that break things down from complex molecules into smaller molecules are going to be catabolic reactions. And the sum of the catabolic or decomposition reactions and anabolic or synthesis reactions is going to be metabolism. And metabolism is going to be everything that occurs in your body that's going to be anabolic or catabolic, and it's all going to require energy. And the energy molecule that we use in our body is ATP, or adenosine triphosphate. And you may recognize from some of the genetic sections that I've given that adenosine is actually a nucleotide that's found in DNA and RNA. However, adenosine triphosphate, so if you take that same nucleotide and you add 3 phosphates on the back of it, you form a high energy carrier molecule. And that's actually going to be called an energy carrier because it couples 2 different types of reactions. And we'll talk about that in just a little bit. But basically the reactions that break things down are generally going to be releasing energy. And the reactions that build things up are generally going to require energy. And the ATP is going to be the carrier molecule that goes back and forth between the high energy form, or the triphosphate, ATP, and the low energy form, or the diphosphate, with a free phosphate in solution. And this is going to have less energy than that molecule. And basically when a reaction that is going to release energy, it would be called exergonic. Usually catabolic reactions are exergonic, and reactions that are exergonic are going to release energy. And the release of that energy is going to get picked up by this ADP molecule and combined with the free phosphate in solution and get stored in that bond between the di and the triphosphate, forming this ATP molecule, which is a higher energy. Then it's going to go somewhere else in the cell, and it's going to help drive a reaction that's going to cost energy cellularly. So an anabolic reaction is generally going to need energy to go into it, right? So energy has to go into a system. It's called an endergonic reaction. And anabolic reactions are generally endergonic. And then also can be a way in which we can take this molecule from one part of the cell to another. We can also store it for later on. So this means that we can take reactions that are going to create energy and reactions that are going to require energy and allow them to have both spatial distance and maybe temporal distance as well. So what type of reactions are we talking about? Well, one of the major reactions that occurs in our cell are oxidation and reduction reactions. And you've always heard of them as redox reactions, and that's because they basically always kind of happen hand in hand. And we'll talk about that as well. That's what's called a coupled reaction. And oxidation reactions are going to be a way in which electrons can actually be taken off of an atom or a molecule or put back on. And when we look at electrons, we're actually going to be looking at these hydrogen ions here, basically, is what's going to happen. And so when we are removing electrons from an atom or a molecule, it's called an oxidation reaction. And this is an example of an oxidation reaction. We basically here are going to be removing 2 of these oxygens. Now that carbon is going to be coupled with a double bond to the oxygen instead of having one of the single bond of the oxygen, with the oxygen being sufficient with the hydrogen here. That hydrogen is removed, so is the hydrogen on the other side of this carbon, and then the carbon is double bonded to the oxygen. That's called an oxidation reaction. And the reverse of that will be a reduction reaction. Anytime that we're adding electrons onto a molecule, so here if we're adding on the hydrogens, for example, here we have now taken this carbon that was double bonded to the oxygen, we've kind of opened it up, so it's now going to be able to bond to the hydrogen that we've added, and this oxygen also binding to this hydrogen that we've added. So we have added electrons to the molecule. And so this is a reduction reaction. Now we have these other types of carrier molecules as well, including NAD and FAD, so nicotinamide, adenine dinucleotide, and flavin, adenine dinucleotide. And these guys are basically able to accept or reduce these electrons back and forth. And these guys are going to be very important because, as I mentioned previously when we talked about coupled reactions, oxidation and reduction are always coupled reactions. And so when a reaction like lactic acid going to pyruvic acid, right? So that reaction is going to be an oxidation reaction. right? And at the same time, NAD+, which is in this oxidized form, is going to end up reduced. So this is a reduction reaction. So we have an oxidation reaction happening at the same time as a reduction reaction, and therefore we're able to have these guys as kind of temporary energy currency, or more along the lines of a hydrogen or electron carrier molecule. All right, and as I mentioned previously, ATP is also going to be an energy carrier molecule. And so sometimes when energy is released during these oxidation reactions, hopefully we try to capture as much as we can, and it's going to be captured by the formation of ATP. Again, that happens from an ADP molecule that has a free phosphate in solution. So here's ADP, there's also a free phosphate solution, and then energy is going to be released from a reaction that's nearby. So an exergonic reaction is going to release energy exits that system, is going to get picked up here, driving this endergonic reaction whereby ADP becomes ATP. Again, this is the high energy version, and really all that energy is stored here in this D to P bond, so from the di to triphosphate. There is some energy here in this mono to diphosphate bond, but not anywhere near as much. So you'll very rarely get it down to AMP, and when you do, you'll see it as more of a AMP molecule is more along the lines of a signaling molecule than an energy carrier molecule. All right, so let's talk about carbohydrate metabolism, because really what happens in your body is you are constantly consuming food or energy that you are then breaking down to use for other reactions. And glucose is one of our major storage molecules for that energy. So carbohydrates are going to need to be consumed and then broken down constantly based on the needs of the cells. Now, some of the needs the cells may have include things like the ATP production, so we may need energy for something else. Immediate energy like ATP. We also might need to create amino acids. We also might need to make glycogen or triglycerides. So we need to create things constantly, and so therefore we need to be breaking down glucose. And when we're not creating these things, we need to be storing that glucose for later. So we'll talk a little bit more at a later date or a later point in this lecture about how we can store our glucose for a later on usage. But let's talk about how we use it if we need to use it immediately. So in order for us to be able to use it, it has to pass through the plasma membrane and get into the cell. And in order to get into the cell, we do this by specialized transporter molecules. We do this by facilitated diffusion, so that means that we are not going to be using any energy. We're still going to be going from high concentration to low concentration, so it's not going to cost us cellular energy to get it into the cell, but it's a large molecule and it's not going to go right on through like something like a pore. So it's going to have to go through a specialized transporter called a Glut or a glucose transporter molecule to be able to bring it in. A molecule called insulin increases the insertion of these transporters, so it increases the amount of glucose transporters that are inside the cell, increasing the rate of facilitated diffusion. Now, you may have heard of insulin in cases of diabetics, right? Diabetics respond abnormally to sugar, and there's a couple of different types of diabetes, but basically what happens is this either goes awry because you aren't making enough insulin, or you're unresponsive to the insulin, and your transporters end up not allowing the right amount of sugar in. Anyway, insulin is involved in sugar uptake. So, what do we have to do when we take in glucose? When we're bringing in that glucose, we're breaking that glucose down for the purposes of creating ATP. And we do that by a process called cellular respiration. And cellular respiration has a lot of different reactions. 4 major sets of reactions, which each have their own subset involved in them, and we're going to talk about them extensively. So you're going to see me coming back to the same diagram a couple times just to kind of bring you back to the overview of the process. You have an idea of where we are, and kind of reorient you before we move on to a next topic, for example. So the very first reaction is the glycolysis reaction, whereby we're taking the glucose molecule, we're splitting it in half to make pyruvic acid. I apologize. So from there, I'm just going to skip forward so you can see what's happening here. So this is the first step, taking glucose, 6-carbon sugar, breaking it into 2 3-carbon molecules. And this does release some energy, not a lot, mind you. It releases about 2 ATP and 2 NADH and 2 free hydrogens. Right? And then, although all of those hydrogens you can consider to be electrons, we're going to talk about the electron transport chain, that's step 4, at a later point. But just picture that the release of these electrons eventually turns into energy. So you can consider this kind of like an investment, where it's not going to be immediate energy, but eventually later on it's going to grow and become more money at the end. All right, so we have two, after glycolysis, we have two pyruvic acid, two of those 3 carbon molecules, and they're going to get turned into acetyl coenzyme A. And the formation of acetyl coenzyme A in and of itself is also going to release some byproducts. So it's going to release some of those electrons. Again, those are important in creating ATP later, so it's not ATP now, but it's an investment in the future. And then we have some more NADH and some carbon dioxide. Well, where did that carbon dioxide come from? You got a picture that we had this pyruvic acid. It was a 3-carbon structure. Now we're releasing individual carbons. So we're breaking it up. So we're going from more structured, right, a 6-carbon ring to a 3-carbon chain to individual carbon molecules. And each time we're doing that, we're releasing energy. But let's get back to acetyl coenzyme A. So that's created after we convert it from pyruvic acid, again, releasing some other byproducts, and it's going to enter into the Krebs cycle. The Krebs cycle is going to have a bunch of stepwise reactions whereby we are releasing more ATP, NADH, FADH, and carbon dioxide as a byproduct of this. This is why we are going to be off-gassing carbon dioxide, because we are constantly breaking down our sugar and ending up with more carbon dioxide in our blood. That's why we have to have the process of respiration, right? And then all of these hydrogens that are released at all of these different stages, including glycolysis, acetyl coenzyme A formation, and the Krebs cycle, are all going to release these hydrogen ions, which you can consider to be electrons, right? Those electrons enter into the electron transport chain, and then they're going to end up creating ultimately either 26 or 28 ATP, and we're going to be using oxygen to do this. This is called aerobic respiration, and what's happening during this aerobic respiration is we are creating water. So our end products are going to be carbon dioxide and water, and we're going to need to bring in oxygen and glucose. All right, so that's kind of the overview of what we're going to talk about. Now we're going to be talking about each one of these piecemeal as we go along. Okay, so glycolysis is that very first step where we're taking that 6-carbon glucose molecule and we're splitting it in half. We're making 2 3-carbon molecules. They're called pyruvic acid. There are 10 reactions. I'm not going to quiz you on every step of these reactions, and I certainly do not expect you to know the molecular structures of every of the byproducts, but I would like you to know stepwise which goes in what order. So right now, again, we're focusing on this upper box here in blue. So this first step, glycolysis, basically we're breaking down a 6-carbon sugar. It's a ring. We're breaking it down into 2 3-carbon molecules, and we're basically going to be taking that glucose. We're going to have to put in a little bit of energy here. So to get this jump started, we actually have to put in energy in order to get energy out. So although it does show this energy coming out, that energy going in is missing in this overview diagram. We have to bump start this process with 2 ATP. So although we get 4 ATP out, we really only get 2 ATP out net because we have to put 2 in to get started. All right, so we go from glucose to these intermediate products. Dihydroxyacetone phosphate, glyceraldehyde 3-phosphates, and those are going to end up, again, releasing energy to create pyruvic acid. And we're doing this stepwise because we don't want to release all the energy all at once. We want to be able to capture as much of that energy as possible in ATP. And so by doing all these intermediary reactions, we're able to slowly, one by one, capture these small interactions and release of energy into ATP molecules. All right, again, I don't want you to focus on all of the biochemistry here, but I do want you to understand that we are taking reactions in a stepwise fashion that are small changes that are releasing energy one step at a time. Okay, so here's our glucose molecule, and then we go from glucose to glucose 6-phosphate. I will never show you this structure and expect you to know the difference, but I could ask you in which order or which comes first. All right, so glucose goes to glucose 6-phosphate. That's basically here. And then if you keep your eye on that, that's going to change here when we switch to fructose 6-phosphate. So we basically change the way the carbon ring is oriented. It's no longer a 6-carbon sugar. Now it's set up in a ring. It's a pentagon instead of a 6-carbon ring. It's still got 6 carbons. I apologize. It's not a 6-sided ring now. It's a 5-sided ring now. All right. From there, we're going to end up putting in ATP. So this is our second time. I may have missed that here. First we put in one ATP molecule for the conversion of glucose to glucose 6-phosphate. And then we're going to have another conversion from fructose 6-phosphate to fructose 1,6-bisphosphate, which is also going to require the addition of one ATP molecule. So that if you're keeping track, that's the second ATP molecule that has gone in. Fructose 1,6-bisphosphate is going to go to these two, right? Dihydroxyacetone phosphate and glyceraldehyde phosphate, which I just spoke of previously in the kind of the overview. And now we're going to start getting our money back out of this investment. So now we're going to end up gaining 2 NADH and 2 hydrogen ions, so 2 things that are going to go into the electron transport chain to bank us money later on. And then we end up getting a 1,3-bisphosphoglyceric acid molecules. We get 2 of those. And then we're then going to end up having 2 ATP molecules come out. So ADP goes in, free phosphate goes in, and ATP, or high energy, comes out. From there we go to 3-phosphoglyceric acid and 2-phosphoglyceric acid, eventually down into phosphoenolpyruvic acid, and eventually into pyruvic acid. And again, we're going to have one more set of ATP molecules. We get 2 more here. So overall, we're going to have put in 1 and 1, so a total of 2, and gotten out 2 and 2, so a total of 4. So 2 in, 4 out, net gain of 2. Seems like a lot of work to only get 2. And that actually is going to be really all we can do until we start adding in oxygen here. So this is basically going to be the difference between aerobic and anaerobic respiration. This is why an organism that's able to use oxygen for metabolism is able to get 34 ATP instead of 2 out of that same glucose molecule. So it was a drastic increase in the ability of an organism to be able to get energy out of the same molecule. Now, if we don't have oxygen, we're going to go into aerobic conditions, and basically what will happen is that pyruvic acid molecule is going to get reduced and it's going to form lactic acid. And this is what happens when you over-exercise and you end up with a cramp. You end up with lactic acid buildup in a particular region because the body is overusing its glucose, and it's basically saying, well, I'd rather get those 2 ATP than nothing. So it's breaking those down without being able to use what's left of that pyruvic acid into the next stages of the cycle, because we basically are making more pyruvic acid than we're able to consume. And then it's going to get converted into lactic acid, and then if it overwhelms the system in terms of waste removal, it's going to end up building up in your tissues. However, under normal conditions, oxygen is plentiful, so that's aerobic conditions. So most of the time, the cells are going to convert pyruvic acid into acetyl coenzyme A. Okay, so I'm just going to kind of bring you back to that original figure. We are now on the step 2, where we're taking pyruvic acid and we are converting it into acetyl coenzyme A. But I want to make something clear here. We have now entered into a specialized organelle called a mitochondria. A mitochondria, also chloroplasts, incidentally, although we won't be talking about them in this lecture, are a specialized type of organism. They have their own DNA, they have their own specialized capabilities, namely that they're able to undergo aerobic respiration, which was a huge advantage for whatever cell ended up encountering it many, many billions of years ago. And so it ended up incorporating the mitochondria into its cells. It's called the endosymbiont hypothesis. So basically, a larger cell ate a mitochondria and decided, you know what, instead of me digesting this mitochondria right now and getting an immediate bang for my buck, I'm going to keep this guy alive and allow it to break down whatever sugar I have and get continuous bang for my buck. And so the mitochondria ended up being protected by the larger cell, and the larger cell ended up having an energy source that was sustainable. So mitochondria are actually very unique, and they have these specialized folds here. They're called cristae. There's only a couple denoted here, but really they're back and forth constantly. And across this is a matrix, and across this mitochondrial matrix, we perform a couple things. And we'll talk about those folds a little bit more when we get to the electron transport chain. But one of the things that happens in the mitochondria is the conversion of pyruvic acid into acetyl coenzyme A. And basically, it's going to go through a couple intermediary molecules. Again, I'm not going to ask you for the molecular structures of anything, but I do want you to know the order of these steps in the cycle. So pyruvic acid is going to go to pyruvate dehydrogenase. This is going to give us a little bit of cellular energy, a little bit of NADH, also going to off-gas some carbon dioxide. Then we're going to end up making, with the addition of this acetyl group that's going to make coenzyme A. And the acetyl coenzyme A is going to enter the Krebs cycle. So that is what's going to be happening in our next step. So again, just to kind of reorient you, we've already gone through the process of glycolysis where we're taking a glucose molecule, it's a 6-carbon sugar, we're converting it many different steps from a 6-ring into a 5-ring, and eventually chopping it into pyruvic acid, which is a 3-carbon molecule. Some of that is going to get converted into acetyl coenzyme A. That happens in the mitochondria. Then it enters into the Krebs cycle, which is what we're going to talk about next. The Krebs cycle is going to release more ATP, also more carbon dioxide. We're also going to get out of it some NADH, some FADH, and also some electrons that are then going to get pushed into that electron transport chain. All right, another term for the Krebs cycle is the citric acid cycle. Like the other ones, it's split into multiple reactions, so a lot of smaller subsets of reactions, and there's 8 of them. Again, it's going to happen in the matrix of the mitochondria. So this one's going to be an overview, and then we're going to talk about the intermediary molecules. But basically what happens is that acetyl coenzyme A enters into this citric acid cycle and becomes citric acid. And in small subsets of these reactions, we are releasing NADH and the hydrogens. Again, hydrogens are going to end up going to the electron transport chain, and that's going to end up being more bang for our buck later on. So they're actually quite valuable, even though right now it doesn't look like ATP. Eventually they become the ATP from the electron transport chain. In each of these reactions, we're releasing some carbon dioxide. Why? Well, we're taking a 3-carbon molecule and we're breaking it down into small carbons, so single individual carbon molecules that are, they're bound to oxygen, but from 6 to 3 down to singles, right, in terms of the carbons. And each step of that is going to release more. We release some ATP, we release some FADH, more NADH. All of this is going to end up going to the electron transport chain to end up creating more ATP for us later on. Okay, take a breath. I'm going to walk you through it. This is everything that we just talked about, except I've added the intermediaries in here. Again, we're taking that pyruvic acid. We've converted it into acetyl coenzyme A. Again, this is all happening in the mitochondria. And that little reaction is going to have released some NADH and hydrogen that's going to go to electron transport chain as well. Then we enter into this citric acid cycle, right? So acetyl coenzyme A gets that coenzyme A cut off of it, and then we end up making citric acid from that. Citric acid goes to another intermediary molecule, that's isocitric acid. You see we're making small changes to these molecules because these small subsequent changes one at a time instead of having one large change means that every time that we're releasing energy, we're able to acquire it from the ATP, well, the ADP, free in solution, and the free phosphate conversion into ATP. But actually in this set, we're making a lot more NAD and FAD than we are of ATP. But I digress. So the isocitric acid gets converted into alpha-ketoglutarate acid, and that's going to release a little bit more of some NADH and some hydrogen. Again, that's all going to go to the electron transport chain. The coenzyme comes back in. Coenzyme A comes back in this intermediary of 4 here and brings us over to acetyl coenzyme A. And then that coenzyme A gets converted off again to make succinic acid. And so you can see that this coenzyme A is actually more of a helper molecule. Even though it does get converted onto and off of, it pretty much remains unchanged throughout these reactions. So basically it targets this to get brought into the whole cycle in the first place. And then it helps convert from, for example, it converts here, it helps convert here from alpha-ketoglutarate acid into succinyl-CoA, and then coming off again into succinic acid. So it's really just helping all of these stages happen, and it's also keeping it cyclical. So we're only going in this direction, right? We're not going backwards because this coenzyme A is going to be used for other reactions elsewhere. So as soon as it comes off, it gets picked back up, and so it's not able to drive the reverse reaction. Again, I digress. So succinic acid, that conversion from succinyl-CoA to succinic acid is going to actually release some ATP, and it's going to do that by an intermediary product. Here, this GDP to GTP, but just picture that works the same way as ADP and ATP. It just happens for a lot faster. It's not as longer term of a storage carrier molecule. ATP is still a short-term carrier molecule, but this one is a flash in the pan. Okay, so from succinic acid, to fumaric acid, which is step 6, is going to release more FADH. Again, that's going to head out to the electron transport chain. Our next thing is going to be malic acid, and this is going to require the addition of some water. So water is going to have to come in at this point. We're also going to be releasing here some NADH. So again, we're picking these energy molecules up one little piece at a time in each of these steps of the reaction, instead of it happening all at once, where it might overwhelm the ability of the system to acquire these energy molecules. Anyway, the end here is going to be the oxaloacetic acid. And then that is going to end up being converted here and help get converted into the citric acid, again with the addition of water and with the new incoming acetyl coenzyme A, thus completing and restarting that Krebs cycle. So after we end up releasing all of these hydrogen ions, which again you can look at them like electrons, right? They're going to end up going into the electron transport chain. And that happens in the mitochondria. And it's basically a series of electron carriers that happen in a row. And each one is going to be first reduced and then oxidized over and over and over. And every time it picks up electrons, right, it's reduced. And every time it gives up electrons, it's oxidized. And we're going to be doing this over and over to release energy. So first we have energy coming in and then energy release, and then energy coming in and then energy release. And every time that we're using that, we're driving cycles that are forming ATP. And again, we're doing that from a DP or diphosphate plus whatever free phosphate is in solution, plus the released energy from these exergonic reactions. Remember, exergonic reactions, energy exits the system. And this whole process of chemical reactions and hydrogen ions being pumped in and out is actually known as chemiosmosis. And that looks a little bit like this. Okay, so this is the mitochondria. And I may have told you that we had cristae, which are these little folds inside this matrix. What I didn't mention is that we had 2 different membranes. And this is because of that endosymbiont hypothesis, or kind of like drives back to that, because the organism, the bigger cell that ate that mitochondria initially, needed to mask it from non-self. So that outer membrane is going to look like whatever organism this mitochondria is in. Whether it's in a human, it's going to have human characteristics. If it's in a different mammal, it's going to have that characteristics of that mammal for that outer membrane, right? But the inner membrane is going to be distinctly mitochondrial. So that inner membrane is comprised of whatever it was comprised of many millennia ago when it was its own organism. And inside this matrix, we have a disparity of hydrogen ions. So inside that inner and outer membrane, we have a much higher concentration than we do inside the matrix. And this is important because it means that we are driving these hydrogens basically in just by going from high to low concentration. So it actually doesn't cost any energy for that hydrogen to go right on through this hydrogen channel. In fact, it actually drives the creation of ATP. So hydrogens come right on in because they go from a concentration gradient of high to low, so they come in no problem, and they go past an enzyme called ATP synthase. And as you might imagine, synth meaning synthesizing and making ATP. So here we have ADP plus free phosphate in solution, and the energy while that hydrogen ion is sliding through that channel is going to drive ATP synthase to make ATP. Okay, and then we have this electron transport chain that I was talking about, which includes specific proton pumps that are going to drive that back out. So we're going to drive that hydrogen ion back out. That's going to maintain that concentration gradient, therefore making that hydrogen ion want to come back through, therefore creating more ATP. And so it's this cycle whereby every hydrogen ion that comes in is going to drive the creation of an energy carrier molecule, and then being forced back out, which is going to use energy from that NADH, which we also acquired earlier from the Krebs cycle. Okay? All right, so the ones that we talked about are not the only electron carriers. So we've talked so far about ATP, we've talked about NADH and FADH. There's also one called FMN, or flavin mononucleotide. There's things called cytochromes, iron sulfur centers, copper ion atoms, and coenzyme Q. So these are all different types of electron carrier molecules. So if you picture that the most common electron carrier molecule is ATP, right? That's our $20 bill if you consider it an energy currency. But we also have $5 bills, we also have $50 bills, right? We have a lot of different other types of currency, and electron carriers are the same way. All right, so let's talk about what's actually happening inside there. So inside the inner mitochondrial membrane, we have these little clusters of these carriers, and each of these clusters has complexes that are going to be a proton pump that pushes those hydrogen out. All right, so that looks like this. Again, here's the outer and inner mitochondrial membrane. So that's that space in between. Here's the inner mitochondrial membrane. Here's the mitochondrial matrix. So now we're inside the mitochondrial matrix itself. And again, we have way more of the hydrogen ions on the outside than we do on the inside. And we have that because we're constantly pumping them out, right? And we do that by a series of different electron transporters. So it's here, for example, it's going to get pumped out. That's going to cost us energy. So here it costs us the NADH, but we brought that NADH in from the Krebs cycle anyway, along with that hydrogen ion. So it basically brought in its own energy to drive this reaction. And then those hydrogens are going to all get kicked out from multiple different types, including cytochrome B, C complex, cytochrome B, cytochrome 1 complex, and a ferrous sulfur center, and all of these are going to be pumping, as I said, these hydrogens out. Then the hydrogens are going to come back in through this nifty little, looks like a playground slide, but it's going to be driving the synthesis of ATP using an enzyme called ATP synthase, which basically just sits here and waits for these hydrogen ions to slide in and uses that energy from that to drive the creation of ATP using ADP and free phosphate that are also just sitting there kind of waiting in the shadows there. So the overview of all of that is called cellular respiration. And just as a side note, this is the exact opposite of photosynthesis, right? Photosynthesis is going to take carbon dioxide molecules and water molecules and create sugars. And it's doing that by taking the energy from the sun. So ultimately, the sugars that we're using were created in plants using the exact opposite mechanism and opposite building blocks. So that's why we need plants so much, because photosynthesis and cellular respiration are basically equal and opposite series of reactions. But the overview of cellular respiration means that for every glucose that we break down, we get between 30 and 32 ATP molecules. So just stop for a second and think about the difference between where we were before we had oxygen, where we had created 2 ATP molecules. We put in 2, we got out 4. We put in 2 more, we got 4 more. That would be such a huge loss in terms of the amount of ATP if we didn't have mitochondria available and we weren't able to utilize oxygen. Because now we're going from, 2 to 30 to 32. So that's an increase of 28 to 30 just for the ability to use this particular other cell, basically, as your energy source, as your biological battery, basically. So the overview of the reaction here, and I know we talked about a ton of intermediary molecules, but really what went in was glucose, right? What also went in? Oxygen, right? We had to breathe in that oxygen, and that oxygen came through the process of respiration, right? And glucose came in through digestion, and then it's going to get compiled and reshuffled through all of those different reactions that we just talked about, releasing subsequently, one at a time, right, in a serial fashion, 30 to 32 ATPs by using ADPs that were found in solution and free phosphates that were found in solution, and these stepwise reactions to end up with byproducts of carbon dioxide and water. So basically, this is what goes in, and this is what comes out. And that's exactly what I mean when I said that this is the equal and opposite set of reactions to photosynthesis, because in photosynthesis, what goes in? Carbon dioxide and water. And then energy from the sunshine becomes glucose, right? And they off-gas oxygen. And that's why we need to have plants around, because they help put the oxygen back into the atmosphere, and they serve as what we call carbon sinks, because they take carbon dioxide out of the atmosphere and store it in sugars. But again, I digress. All right, so let's step backwards again and kind of just talk about the whole thing. So here we are with glucose. We have one glucose molecule, and in the cytosol, which means not inside the mitochondria, just inside the cell itself, the first step happens, and that's glycolysis. Again, we're going from a 6-carbon to 2,3 carbons, and we do that by a whole series of reactions, but eventually that pyruvic acid has two different pathways. In the absence of oxygen, it gets converted into lactic acid, and that's the end of our game, and that's all we get. We get 2 instead of getting 32. So it's quite a major difference. What we can do once we toss that pyruvic acid into the mitochondria. Inside here, we're going to get it converted into acetyl coenzyme A. Again, that releases a little bit of NADH and some hydrogens, which we're going to use in the electron transport chain in just a minute. And then that acetyl coenzyme A is the introductory point to the Krebs cycle, which just goes round and round and round and constantly is producing NADH, FADH, carbon dioxide, and the hydrogens. And all those hydrogens that are created from all of these different steps in the pathway get used to drive the electron transport chain, which is perpetually using multiple different sets of electron carrier molecules to create ATP and free water. All right, that was a mouthful. This is an overview of everything that we just talked about in terms of the different steps, right? Glycolysis, acetyl coenzyme A creation, electron transport chain, Krebs cycle, etc., and their yields of ATP per molecule of glucose. And the yield obviously is overall, as we talked about glycolysis, the yield was actually, you get 4 at the end, right? But you put 2 in, so it's a net. So we get 2 ATPs net. And then we also are going to create 2 NADH plus hydrogen ions, which later on are going to turn into 3 to 5 ATPs, depending on how it's used. And so when you see us say 30 to 32 ATP molecules, this is that variable region, the production of the NADH plus the hydrogen, that can be used during 2 different pathways. Again, this is another topic. I mean, the same pathway, but 2 different ways it can go throughout that pathway to create either 3 or 5 later on. So this is an investment that sometimes yields 3 and sometimes yields 5 ATPs. When we head into the formation of acetyl coenzyme A, that's going to create 2 NADH and 2 hydrogen ions, and then again, remember, that's not going to be an immediate yield, that's going to be a yield later on after the electron transport chain of 5 ATPs. And then the Krebs cycle itself is also going to yield in multiple different stages, and it's shown in that cycle which stages they are, but the major stages of the oxidation of succinyl-CoA to succinic acid is going to yield 2 GTPs. Remember, those are a flash-in-the-pan intermediary molecule. They're immediately converted into ATPs. So it's an immediate use of that ATP. That ATP is immediately available. And then these guys here, also production of 6 NADH and 6 hydrogen, and production of 2 FADH. And all of that is later on going to have to go through the electron transport chain, but ultimately is going to yield 15 ATPs and 3 ATPs for a total of between 30 and 32 ATPs per glucose molecule. Again, we only got 2 if we weren't using oxygen. So this is why aerobic respiration is so incredibly vital. But sometimes we have more glucose available than we need. So what do, right? We want to store it. We don't want to waste it. We want to make sure that later on, if we do need it, we have access to it. So we store it as something called glycogen. And the creation of glycogen is glycogenesis, right? So genesis, creation, glycogenesis, creation of glycogen. And if we are breaking glycogen down, right, lysis being broken down, we are using a process called glycogenolysis. And we've talked about this a couple of times. I know when we were talking about digestion, we talked about this. But basically, glycogen can be broken down into glucose to be able to get consumed for cellular respiration purposes anytime the body needs ATP. All right, so this blood glucose level, you see all these different arrows that can go different directions, and we can have the glucose can get stored in the glycogen in this direction if we have more of it in the blood than we need, and it can go that direction if we have less of it in the blood and we need to break things down. So basically the red arrows are in a situation where we have more glucose than we need and we need to store it, and the blue arrows are when we need to break down glycogen to be able to create a higher blood sugar levels. Right, so glucose is going to get converted into glucose 6-phosphate and glucose 1-phosphate. Again, this first step is going to cost some money, so it's actually going to cost us some energy to be able to take glucose and store it as glycogen, but it doesn't cost any money to take it back out. So usually the creation of larger molecules is going to cost energy. Remember, we talked about anabolic reactions, building things up, and usually those are endergonic because energy has to go into them. So this pathway is going to need energy going in. But ultimately we're going to make glycogen, And then if we wanted to be able to take that glycogen and break it back down, it's not going to cost us any energy to take the money back out of the bank. So breaking glycogen, which is a larger molecule, down into glucose is not going to cost us any money. But we are going to do it by a series of pathways. So a lot of things in metabolism are going to be stepwise reactions, because you don't want to just go directly from A to B, because it's really easy to go back from B to A. So you want to go to intermediate 1, intermediate 2, intermediate 3, and therefore in order to undo it, you have to go back stepwise through those same reactions. Then again, those intermediate molecules here, so you can just read them, glucose 6-phosphate, glucose 1-phosphate, and uridine diphosphate glucose. Now, at the bottom here, it's telling you what stimulates glycogenesis or glycogenolysis. And glycogenesis is actually stimulated by insulin, so that goes back to the topic that I just kind of touched on for a second about diabetics and how they may not be responsive to insulin properly, so they might have issues with glycogenesis. And glycogenolysis is actually going to be stimulated by glucagon and epinephrine, which makes sense, right? Because if you think about epinephrine, that's that fight or flight response. You might need sugars during a surge of adrenaline. And so we're going to be breaking that glycogen down into sugars that we can immediately use for fighting off an attacker or running away if someone's trying to hurt us. Okay, so we can also make glucose from other things. So we can have different biomolecules converted across different pathways. One of the places where we can take from to be able to create glucose is proteins. So we can use proteins to be able to form glucose, as well as using parts of triglycerides, not all of them, but the glycerol portion of triglycerides, lactic acid, and also amino acids. And it's not surprising that we can use lactic acid to make glucose, right? Because we took glucose and broke it down into pyruvic acid and then got that converted into lactic acid just a second ago. So you can see how easy it might be to go the opposite way, take lactic acid, make pyruvic acid, and make glucose. And you can also use amino acids to be able to build this glucose molecule. And any of the ways that you're doing these pathways are called gluconeogenesis. And genesis being creation of, neo being new. So we're making new sugar here. We're not taking glycogen and breaking it down. We are taking other molecules and using them to create new glucose. So gluconeogenesis, creation of new glucose molecules. And lots of different things are going to stimulate gluconeogenesis, specifically cortisol, glucagon, and different thyroid hormones. So having a thyroid disorder may end up having issues with sugar balance as well. All right, so this is basically showing you different gluconeogenic pathways. And gluconeogenesis, again, it's stimulated by things like cortisol and glucagon, and it can come from multiple different pathways. Now, it's not surprising that it comes from pyruvic acid and goes to glucose, because the opposite direction that we just talked about was glucose being broken into pyruvic acid and then being either sent into the Krebs cycle or converted to lactic acid. So it's also not surprising that lactic acid can get converted into pyruvic acid and enter into this pathway from that direction. Right, because any pathway that we take one direction, we can take the other way, but we have all these intermediate steps, so we want to make sure that we have to go through all of them to be able to have that happen. Also, certain amino acids can be used, and glycerol from the triglycerides can be used. All those can be used to drive the creation of new glucose molecules. Again, that is called gluconeogenesis. All right, so we just talked about carbohydrate metabolism, so let's talk about lipids for a second. Remember, lipids are going to be nonpolar. They're hydrophobic, right? Picture oil, bacon grease. So they're not going to dissolve in water. But blood plasma is almost all water, 90% water. And we need to get these through the blood plasma into circulation. And so in order to do that, these lipids are going to have to be transported with specialty proteins that are called lipoproteins that are specifically produced in the liver and intestines. And we talked about this when we talked about our digestion section, so this should be something that you're a little bit familiar with. Now, the term amphipathic, I'm not sure that we have gone over yet, so let me touch on that before we go anywhere else. Amphipathic means that it has one side that is polar and one side that is non-polar. So here you may recognize that these are phospholipids, right? These little blue ones. And the same thing that you see when you look at a phospholipid membrane, for example, except when you look at a membrane, it's a bilayer. This is only a single layer. This is called a micelle. Basically what happens is all of these orient themselves such that the heads which are polar and able to interact with water just fine, are all oriented in one direction, kind of like a pincushion, whereas those tails, which are hydrophobic, are all oriented in the other. So basically what I'm saying is an amphipathic molecule has one side that is polar and able to interact with water, and one side that is nonpolar and able to interact with oils or lipids. And if we put a whole bunch of these together, we end up with this micellular structure, and they can get larger or smaller depending on what they're carrying, but inside they're going to have lipids or oils or something that doesn't want to interact with water, right? Some sort of nonpolar molecule. And then we're going to be able to interact well with the outside environment because we are masking the fact that we have lipids on the inside by using a bunch of amphipathic molecules that have polar heads that are going to interact well with the watery environment. All right. So we have a lot of different types of lipoproteins. And you may have heard of these when you've heard about your blood cholesterol. One of them is chylomicrons. Chylomicrons are basically going to be when we are taking lipids and storing it in fat cells. So this is basically a storage, not a storage unit because it's sending it over to the storage unit, but a trucking system to send something to a storage unit for fat. So adipose tissue is where we're going to store our lipids. We also have VLDLs, LDLs, and HDLs, and they all have to do with the density of the lipoproteins in a centrifuge. So basically, if you spin them down, they are in different layers. But functionally, they're different because they transport different things, and they are better or worse for you. So let's go through them. Very low-density lipoproteins are going to be responsible for transporting triglycerides through your system. From the hepatocytes, remember your liver is going to be responsible for pulling in a lot of these guys, because you might also remember that these guys have to get sent through like the lymphatic system because they're not going to be able to go directly into the bloodstream. So they're going to get absorbed through the intestines and then get sent in through the hepatic portal vein and then eventually end up in the system that direction, and that's how they're going to get transported to adipocytes, right? We also have low-density lipoproteins. These are LDLs. That's about 75% of the cholesterol in your blood, and they're going to be responsible for delivering the cholesterol to the different cells. Remember, cholesterol is going to be very important for the cell membrane, structures, and so we do need cholesterol in limited amounts. Obviously, with everything, it's all about homeostatic conditions, right? Now, high-density lipoproteins, or HDLs, are going to be taking excess cholesterol from the body cells and the blood and transporting it to the liver for elimination. So as you might imagine, it's kind of like a battle between your LDL and your HDL. These guys aren't really playing a role in terms of having issues with your cholesterol. But when we say good cholesterol and bad cholesterol, right, the LDL is dropping cholesterol off at the cells, and the HDL is removing it from the cells and eliminating the excess. So it's a ratio, right? And you want to make sure that you don't have the wrong ratio, because then you're dropping too much off in the cells and not picking enough up. Or you could be picking too much up and not dropping enough off. So really you want to make sure that it's a ratio of the LDLs and HDLs that are within the correct homeostatic conditions when you talk about cholesterol imbalances. So cholesterol comes from exogenous sources. You can eat it in things like eggs, different types of organ meats as well, like liverwurst, also dairy products. But most of your cholesterol is actually synthesized in your body by hepatocytes, so cells in your liver. An overall cholesterol level is associated with greater risk of coronary artery disease, but again, it is about a ratio of the LDL and the HDL. And we're able to deal with cholesterol levels by multiple different routes. One, changing your diet. Don't bring in any more cholesterol than you need to. Increase your exercise, because making sure that your exercises is going to jumpstart your metabolism, and also certain drugs if we have to get to the point where you use drug intervention to be able to reduce your cholesterol levels. In fact, there's a ton of cholesterol drugs out on the market. Now, as I mentioned previously, you can actually take your lipids and break them down and use them to help make triphosphates. You may remember that your lipids have that phosphate head, right? And triphosphate has a P right here, so as you might imagine, it's the same or similar molecule that is going to be needed to be able to produce the ATP. And if we don't need the lipids that we bring into our body, we end up storing them in adipose tissue. So it's going to be stored in your fat cells. And fun fact, it's a lot easier to store them than it is to break them down. So different lipids can also be used structurally, or we can use them to synthesize other essential substances that we may not have ingested enough of in our body. All right, this is a list of different types of lipids that are found in the body. Some of them that you may recognize, some of them that you may not. So fatty acids are going to be the main component of triglycerides and phospholipids, which are going to be, again, a phospholipid bilayer in your membrane, etc. They can also be broken down to make ATP, right? Because, again, that P is going to be used. The triglycerides are also a different type of lipid. They're involved in insulation. So it's like whale fat, for example, blubber, energy storage as well. So this is, again, going to be fats and oils. So when a bear goes into hibernation, they're going to come out with a lot less fat because they're going to have used that energy over the winter. Phospholipids, as we just talked about, those are the lipid components of the cell membranes. And those are the ones that are going to be the amphipathic molecules. In a cell membrane, it's a bilayer. In that micelle that we just saw, it's going to be just a single layer. Multiple different types of steroids. Including cholesterol, which is a steroid as well. And steroids, like cholesterol, are found in all of your animal cells. We also are going to have sex hormones, which are which are steroids as well, like cholesterol. And so sex hormones are circulating sometimes during different times of reproductive cycles, and they're going to stimulate different reproductive functions and sexual characteristics. Some other steroids, so that's what you think of when you think of steroids, you think of like testosterones, right? But some other steroids include vitamin D, for example, which is used for bone growth, or adrenocortical hormones, which are regulating your metabolism and salt and water balance, etc. Things like bile salts, which we talked about extensively when we talked about digestion, but they're actually helping to absorb the dietary lipids to be able to get them into the circulation eventually. Some other types of lipids, these guys you probably haven't heard about before, these echinosoids. And these are, although we probably did talk about them a little bit when we talked about our immunology, but they are going to have a response to hormones, blood clotting, inflammation, immunity, right? The secretion of different things in the stomach, like stomach acid secretion. Also, they're going to be a response to different stressors, so they will help with smooth muscle contraction and airway diameter, and then also the breakdown of different lipids. Some other lipids you may recognize include vitamin E and vitamin K. Vitamin E, you might have used it on like a scar, for example. It's known to promote wound healing and prevent scarring or fibrosis. Also an antioxidant. Vitamin K is needed to help synthesize specialized blood clotting proteins. We also have things like carotenes, which is used to synthesize vitamin A, which is why Bugs Bunny eats his carrots, which is interesting because eating the vitamin A has actually been demonstrated to be very important for vision, and it can end up being reduced in, for example, you can end up with a prenatal issue of low amounts of vitamin A during pregnancy or in third world countries. So you can end up with night blindness if you have a lack of vitamin A. It's actually a really cool story from the GMO perspective because it's one of the first things that they were able to make and distribute to third world. So they ended up making golden rice. So therefore people who were malnourished but were still eating large amounts of rice were able to get normal levels of vitamin A and therefore able to have vision, and they've eliminated pregnancy and night blindness. But I digress. Those are all different types of lipids that you might find in your body. Okay, so adipose tissue is a storage site for triglycerides and chylomicrons and very low density lipoproteins. And that's about 98% of all of the energy reserves in our body are actually going to be found in adipose tissue. And the breaking down or building up of lipids is just like you might imagine, right? Lipolysis is the catabolic pathways, lipogenesis is the anabolic pathways. So breaking things down, lipolysis, building lipids up, lipogenesis. And when we break lipids down, what do we get? Well, we break fatty acids, we break triglycerides down into fatty acids and into glycerol. And we can also synthesize lipids using either glucose or amino acids or whatnot. And this usually is what happens when we're consuming more calories than needed, right? So all of the food that you're eating, you're not drinking fat itself, but it's getting converted into lipids when you end up with too much sugar, etc., in your system. All right, so this diagram is depicting that we have a lot of different ways in which these biomolecules can kind of cross from one type of a biomolecule to another, right? So for example, amino acids would be in the protein section, glucose, these are sugars, right? Glycerols, these are fats and oils, and all of them can kind of go back and forth across one another depending on what the body needs. So for example, certain amino acids can be used to end up with lipogenesis, so the creation of fats, and that's all stimulated by insulin. Or we can break fats down, lipolysis, and again, that's stimulated by epinephrine, norepinephrine, so all of those flight or fight response molecules, also cortisol, so some stress hormones. All of that can help us break down the triglycerides into the fatty acids and the glycerol. And then the glycerol can get sent into the glucose pathway the same way that we turn it into triglycerides in the first place when we ate too much sugar and it got converted into glycerol. So the excess sugar that doesn't make it into these pathways is going to go over into glycerol. Glycerol then gets stored into triglycerides. So all of these pathways can kind of go back and forth, and again, there's a couple intermediaries, and in this way, if you end up deficient in one type of food molecule, you can typically end up compensating for it with other things that you have in the body. Okay. All right, so digested proteins. We haven't talked about protein metabolism yet, so let's chat about proteins for a second. So what are proteins made of, right? Amino acids. So if we break a protein down, we're going to end up with amino acids, and we're not going to store these amino acids. We're going to either reuse them, which is generally what we do, so we're going to use them to synthesize new proteins, or we can oxidize them to produce ATP if necessary, if we need more ATP in our system for whatever reason at that point in time. A lot of different proteins are enzymes. In fact, all enzymes are proteins. Now, not all proteins are enzymes, but all enzymes are proteins, and they're involved in all sorts of processes in the body. These guys are the actual cellular workers. These are the blue-collar workers of the cell. So they're involved in transportation. Some of them are antibodies. We have them in blood clotting effects. They're hormones. So we've talked about a lot of different types of proteins throughout this semester. They're also found in muscle. So proteins are going to be really important, and converting one protein to another is also going to be very important. Specifically, what happens when you eat another animal's protein, even though you might want similar muscle protein, you don't want cow muscle protein, you want human muscle protein. So you have to digest the cow protein down into its amino acids to use those as building blocks to build back up the human proteins. So generally, digested proteins are used to synthesize new proteins. All right, so these are just several different types of proteins, but please keep in mind that there are hundreds of thousands of proteins in the cell. I mean, that might be an exaggeration, but it wouldn't surprise me if there were 50,000 different types of proteins. Proteins do everything inside the cell. So these are generalized types of proteins, and we have some examples, but these are in no way is this list exhaustive. All right, so different types of proteins include structural proteins like collagen and keratin that's found in your bone or in your skin. Basically anything that's going to be structural and able to make a framework of different parts of your body. Also, regulatory proteins, is there anything you hear as hormones or going to be part of any of the different endocrine system responses, different physiological processes like growth and development, anything that's a neurotransmitter, or responses of the nervous system, including insulin, which we just talked about, and responsible for regulating blood glucose level. So regulatory proteins are going to be very specific on the pathways that they control as well. Contractile proteins are basically going to be your muscles, going to be in your muscles, going to allow your muscles to tighten down and shorten, which is going to allow us motion, essentially. Some examples of that include myosin, actin, tubulin. Some immunological proteins, we've talked about several different sets of antibodies and interleukins that are responsible for helping our bodies stand strong against foreign invading pathogens. Other proteins are involved in transport and take substances from one spot to another. The most classic example of this is hemoglobin, right? Hemoglobin is responsible for transporting oxygen and carbon dioxide so that we can basically deliver the groceries and pick up the trash from all of the cells in our body. And then last but not least, we have enzymes. And enzymes all have specific biochemical reactions that they regulate, kind of like a lock and key mechanism. Some simple examples include amylase, which is in our saliva, and that's going to help break down amylose, which is a sugar. It's one of the very first things that you're going to be breaking down in terms of digestion. Basic enzymes are going to regulate specific biochemical reactions, and they're going to be very specific to the reactions that they regulate. All right, so if we're breaking proteins down, we can make several things. Generally, we're going to be making amino acids. That's going to be the first step, but we can convert those into other amino acids, for example. So we have 20 different amino acids, and some of them we simply cannot synthesize ourselves. We have to either ingest from other organisms, so those would be essential that you have to actually eat, but some of them we can convert from one amino acid to another. We can also make fatty acids, ketone bodies, and glucose from proteins. Another thing that we can make out of those amino acids would be ATP. We do that by the Krebs cycle. We just talked extensively about that. And the synthesis of new proteins basically is creating a new protein from the building blocks of the old protein by binding together amino acids in a space called ribosomes. Okay, so this is an overview of where all of the different amino acids can enter into the Krebs cycle. I am not going to go into it, but basically you'll see that all of the different proteins, there are 20 different amino acids, all of the different amino acids are able to enter into the Krebs cycle at different locations and get converted into / back and forth from one to another. All right, this is its own lecture. So I am going to go into it, I'm going to gloss over it, but if you have serious questions about this, we will refer you to my protein metabolism lecture, which will be up shortly. All right, so let's talk about what's happening here. Just to give you an overview of the different things that we are going to be seeing here, this here is mRNA, this long chain here. The mRNA is comprised here of nucleotides. These nucleotides are split up into sets of 3. Those sets of 3 are called codons. And each codon matches a specific anti-codon region on this blue guy here, which is a tRNA molecule. Okay, so we have the tRNA molecule here, which is an anti-codon region that matches up with the codon on the mRNA molecule. That's there. This complex gets started first, and then the ribosomes come in around it. So ribosomes have a small subunit and a large subunit. The small subunit is going to sit on the bottom, the large subunit sits on the top, and they're going to encapsulate the tRNA binding to the start codon. So first, the small subunit is available, the mRNA sits on top of it, and the anticodon is just kind of free. Well, the tRNA with the anticodon is free in solution, and it comes and it binds. Once it binds, we initiate the reaction. Now the large subunit comes and sits down on top, and the whole process is going to start. All right, I'm going to draw your attention here now to this little green ball. These are different amino acids. You can see we have a lot of different colors. We have 20 different amino acids. You won't be expected to memorize them or how they come together, but basically just recognize that they're going to bind to one another in a specific order, which creates a specific structure, which has a specific function. All right, so the first one to come in is going to be methionine. This tRNA with the specific start codon region always brings in methionine. So the beginning of every single protein is going to be the amino acid methionine. Once we have everything in place, let me do one more thing. We have 3 different sites on this large ribosomal subunit. We have the entry site, which is A, and the exit site, which is E, and then the actual site of protein synthesis, which is the P site. This is where the tRNA carrying the methionine and with the anticodon that's going to bind to the start codon is going to sit, is in the P site. All right. So now we have that first one ready to go. Now what's happening is we're reading in this direction. So this is going to be the next one, red. This codon, 1, 2, 3, whatever it says, is going to match up with another anticodon on a different tRNA that's going to come and bind in this A site. And that tRNA with its anticodon is going to have a specific amino acid. So the tRNAs are a way that we can convert from the language of nucleic acids into the language of amino acids. And we do that at a rate of 3 to 1. Because this codon is a 3, and we only get 1 amino acid out of it. So if we had 300 of these in a row, we would only get 100 of the amino acids. Actually, that's not true. We would get 99 because the stop codon does not code for an amino acid. That's a trick question, but I'm off topic. All right, so this anticodon is going to be specific for this codon for whatever the second amino acid is going to be. It comes in and binds. Boom. Now, this original methionine, which was on tRNA 1, is going to get transferred over to amino acid 2. At that point, this whole sequence is going to shuffle over one. Now this one is able to go free into solution, and this guy here, who is now holding a chain of 2, is left in that P site. That process is going to lather, rinse, repeat until we reach, so this is going to be a growing chain, until we reach a stop codon. When we reach a stop codon, the whole thing is just going to disassemble, and the protein is going to be left free in solution. So basically, here's the overview. We have the the small subunit, tRNA binds, the large subunit binds on top, and it moves along this mRNA molecule as the protein is growing out of the back of it until it reaches the stop codon, whereby the whole thing disassembles. The tRNA, the large and small ribosomal subunits, the mRNA, and the complete protein are all released. And then the ribosomes go and do this again with another mRNA, and the mRNA can continue to do this until it is degraded. So that is a quick and dirty overview of protein metabolism. I apologize if that seems like I tossed a lot out at you at once, but it is what it is. That was a very meaty slide. All right, so previously I talked about the fact that molecules can have kind of crosstalk at these different crossroads, these metabolic crossroads. And molecules can basically go back and forth. And we have a lot of very specialized chemicals in these cells that are very important to these metabolic crossroads. That's going to include things like glucose 6-phosphate, pyruvic acid, acetyl coenzyme A. These guys are going to be key factors in moving forward or going backwards in these metabolic pathways. So glucose 6-phosphate is important because it's involved in synthesizing glycogen, also in releasing glucose into the bloodstream and creating nucleic acids and glycolysis, I apologize, so breaking down glucose as well. And pyruvic acid, I've said probably 100 times in this lecture alone, so you are very familiar with the fact that it's important in the production of lactic acid, for example, if we're doing anaerobic respiration. We can also use it to produce alanine, which is an amino acid. We can use it to create new glucose molecules, right? And then we can also send it into the next stage, right? As we can turn it into acetyl coenzyme A and send it into the Krebs cycle. Acetyl coenzyme A is important in helping any of the 2 carbon acetyl groups enter into the Krebs cycle. So remember, we lost that 1 carbon molecule, like carbon dioxide, so we went from a 3 carbon chain down to a 2 carbon chain. And these 2 carbon chains with acetyl groups on them are going to enter into the Krebs cycle. Acetyl coenzyme A is also involved in the synthesis of lipids. So these are very important metabolic molecules. All right, this is everything that I've just told you guys. So most of this is stuff that you should already have seen before, and yes, I know we have a lot thrown together on the same page, but let's walk through it. All right, so we have glucose that in the blood can go back and forth between glucose and glucose 6-phosphate. Glucose 6-phosphate is one of those molecules that can go multiple different pathways, right? It can go to make glycogen, for example, right? It can go to make pyruvic acid, for example. Pyruvic acid can go on to it could go on to make acetyl coenzyme A and then into this Krebs cycle here, right? We can also have anaerobic reactions where we go and make lactic acid or alanine. So all of these molecules have a lot of crosstalk in what they are able to do, right? And in this way, if we have a shortage of something, we'll probably be able to draw on it from an alternate molecule that we are able to use in a pinch. This is an overview of the different metabolic processes. So basically, this one is glucose catabolism. This is when we are breaking down glucose, and that's called cellular respiration. And this is the biggest one that we hammered home this entire lecture. This is where we're creating ATP from. 4 major steps include glycolysis, Krebs cycle, electron transport chain, and then also the acetyl coenzyme A creation. And if we take one molecule of glucose and run it all the way through, it's going to yield us either 30 or 32, depending on that one step, 30 or 32 molecules of ATP. Right, that's the overview. That overview is going to be split up again into 4 different sections. The glycolysis itself is the very first step. We're taking glucose, it's a 6-carbon sugar. We go through a couple different reactions, and then we create pyruvic acid at the end. Remember, pyruvic acid is a 3-carbon chain. We also have the Krebs cycle and the electron transport chain. The Krebs cycle is a series of redox reactions where we have a bunch of coenzymes, specifically NAD and FAD, and they're going to pick up the hydrogen ions that are released and the hydroxide ions that are formed from different organic acids, etc. Also going to have some ATP produced, and the byproducts of this are going to be carbon dioxide and water. Please remember, this is an aerobic respiration. We require oxygen for us to enter into the Krebs cycle from pyruvic acid. Otherwise, we're just going to end up going into lactic acid, and that's going to be it, folks. All right, so the electron transport chain is our last stage where we're going to get the most bang for our buck. Basically, this is our 3rd set of reactions. Another series of redox reactions where they go through a series of electron carriers. They're basically passed from one carrier to the next. We're going to make a lot of ATP here. Again, we're going to require oxygen for this. And we're going to make that ATP out of, like, that NADH is going to go in, that FADH is going to go in. So we're going to make that ATP from the resulting products of the Krebs cycle. All right, so that's how we break glucose down. But if we have too much glucose in our system, more than we're able to use in cellular respiration, then we have to be able to convert it. So what do we do with it, right? So we take glucose and we convert it into glycogen. That's called glycogenesis. We do that for storage, and we're not going to use it immediately. We can also take it the opposite direction, so glycogen can be reconverted into glucose. That's called glycogenolysis. So making glycogen, glycogenesis, breaking glycogen down, glycogenolysis. We can also create glucose, and we do that using amino acids, glycerol, lactic acid, etc. And again, lactic acid isn't surprising because it just goes right back to pyruvic acid, which then goes right back to glucose. And that's called gluconeogenesis, is the creation of new glucose from amino acids, glycerol, lactic acid, etc. All right, so that's a summary of carbohydrate metabolism. Let's talk about lipid metabolism for a second. So for lipids, we can either break down or build up triglycerides. So how do we break them down? We break them down into glycerol and fatty acids. So those are basically what we're going to have once we split them in half. Well, in thirds, really. And then glycerol is going to be converted, or can be converted, into glucose as well. Again, that's a process called gluconeogenesis. We just talked about that. And it can also be catabolized, and we can do that by glycolysis. And basically, what we can do there is break it down. Fatty acids are going to be catabolized using beta oxidation, and we create acetyl coenzyme A, and you may remember that's very important for entering the Krebs cycle. It's how we're going to be able to create ATP production, and if we end up with too much of those fatty acids, we can convert them into ketone bodies. This is a process called ketogenesis, and this is actually something that is going to be a condition, so it's something that is an abnormal situation if we end up with too much of these ketone bodies. All right, so how do we build up these triglycerides? How do we create them? So we can synthesize them from glucose and fatty acids, and that's called lipogenesis. And when we synthesize them, we then store them. Typically, we're going to store them in adipose tissue. Now, proteins are broken down and built up constantly because this is part of digestion, is breaking things down into smaller pieces. So the amino acids that we are breaking down from these proteins can be sent into the Krebs cycle after what's called deamination. And that's can end up with ammonia, and we can convert that into urea and then send it into the blood and send it out into the urine. So if we end up with a lot of protein being broken down in our body, if we're on a high protein diet, we can end up with a large amount of urea in our bloodstream, and also in our liver and eventually in our urine. We can also convert amino acids into sugars if we need to. That's called gluconeogenesis again. We can also convert them into fatty acids or ketone bodies. And when we create proteins, that's going to be directed specifically from DNA making RNA, and RNA make protein. And I kind of went a little bit into the mRNA about how we're going to be making protein, but that is, again, that's a general biology, and it's an entire lecture. All right, so regulating all of this is a really tricky situation, and it depends on a lot of different chemicals and different signals that come from the nervous system and from the endocrine system. And a lot of metabolism actually is going to depend on how long it's been since you've last eaten, right? So you can kind of jumpstart your metabolism by giving yourself a small amount of food and then getting yourself primed. And so the absorptive stage is basically when we have a lot of glucose available. So you're absorbing glucose in through your diet. And this is what's happening when we have a lot of glucose available. So first of all, it's going to end up coming in through our gastrointestinal tract and ending up into our bloodstream, which is what they're depicting here. Okay? High glucose in our bloodstream can do a couple of things. One, it can end up being stored, right, in the form of glycogen in our muscle. Right? It can also go through oxidation in our tissues, which is really what happens during cellular respiration, right? Making carbon dioxide, ATP, and water, and we just talked extensively about that in the beginning of the lecture. It can also get converted into triglycerides, right? Or it can go through the liver using the hepatocytes and get converted into fatty acids, right? And also some of the amino acids that we're going to pick up from our gastrointestinal tract, those can be stored in proteins, again in our skeletal muscle. And they can go into our hepatocytes and get broken down into fatty acids, or reused as different proteins, or broken into triglycerides and then stored again in adipose tissue. So you can see a lot of these molecules can kind of go both ways, right? When we have a plethora of them, we're going to bring them in and store them. And when we have a famine, we're going to be able to break them down and take them from our reserves. Okay, so we want to be doing a lot of different things. We want to be facilitating glucose and transporting amino acids and making glycogen and creating proteins and also creating and breaking down lipids. So we're doing a lot of different things constantly, and it's all got to be regulated on a global scale. And we do that by hormonal regulation. So all cells are going to be picking up glucose. So that's going to be happening in most cells, and that happens by insulin. So you'll see a lot of these guys are actually circulating hormones, like insulin ends up in your bloodstream, and so it can affect a lot of different cells in the body. Also, most cells are going to be actively transporting amino acids in. Again, insulin is helpful in upregulating the response to that. It's also responsible for the creation of glycogen, so a glycogenesis, and that happens in the liver and the muscle. Protein synthesis is going to occur in all body cells as well. Again, it's stimulated by insulin, thyroid hormones, and insulin-like growth factors. So you guys starting to see a pattern here? All of these are regulated. A lot of these are regulated by insulin. Lipogenesis, which is, again, the creation of lipids or triglycerides. That's going to happen in adipose cells generally and also in hepatocytes, so it can happen in the liver as well. And it's also regulated by insulin. And all of that was during the absorptive stage. Now, the post-absorptive stage is when we don't have food coming in, and the fuels that we're going to be using for all of our metabolic needs are going to come from inside the body already. So that makes it a little bit more complicated, but basically what you're seeing is the reverse of all of the pathways that you just saw when you had a bunch of sugar in your bloodstream, right? So if we end up fasting or starving, our muscle proteins get broken down into amino acids and enter into the bloodstream, right? Those amino acids can then get transported into the liver, and they can be used to create ATP. They can be used to create sugar, which is important because at this point, if we don't have enough blood sugar, we're starving right now. We want the blood to get back, sugar to get back into our bloodstream. So we can be using the breaking down proteins, turning them into amino acids, converting the amino acids into glucose, and then putting that back into the bloodstream, right? We can also use adipose tissue, so that's another source. We can take triglycerides, break them down into glycerols, fatty acids, get them in the bloodstream, feed them into the liver, and use them to create glucose. Also from the liver, we have storages of glycogen, which we can use to create glucose. Also, we can take whatever lactic acid might be building up in our body, and again, that might be coming from pyruvic acid, right? And we can use that to make glucose. So we have a lot of different ways in which we can either create new or convert from other molecules to create glucose itself, right? And then all that's going to get into the bloodstream eventually. So there's a lot of different pathways whereby things can be converted from one set of molecules to another. So glycogenesis is the breakdown of glycogen, right? Lysis being breaking down glycogen. And that's found in hepatocytes and also skeletal muscle fibers, and it's going to be stimulated by glucagon and epinephrine. So the post-absorptive state is going to be regulated by different hormones than the absorptive state, right? Lipolysis is the breakdown of lipids that happens in the adipocytes, which makes sense because that's where the lipids are stored, and it's under the regulation of a lot of hormones you might recognize that are part of fight or flight, like epinephrine, norepinephrine, cortisol, right? And different thyroid hormones as well. And the breakdown of proteins is basically regulated by cortisol, and it happens in almost all of your cells, but particularly in your skeletal muscle fibers. These are going to be muscles that are tissues that are going to need a lot more fuel, basically. Gluconeogenesis is when we are making glucose from something that's not a carbohydrate source in the first place, so not from glycogen. And that happens in the hepatocytes and in the kidney. So basically in the liver and the kidney, that's regulated by glucagon and cortisol. So the body has to respond to basically feast and famine. Although we do eat on a normal schedule of about 8 hours of a time, the body can last for much, much longer if it makes metabolic changes necessary for survival. So fasting is when you go without food for several hours up to a few days. So 24 hours without eating would be considered a fast, even 48. I'm not sure exactly where starvation comes in, but starvation happens when you go to the point where you have inadequate food intake for weeks or months, which definitely is going to end up in malnutrition. Okay, so not something that's going to end up where you're just depleting your reserves, but you've gotten to the point where you don't have enough left to be able to meet the body's needs. That's considered starvation. And one of the first metabolic changes that happens both with fasting and starvation is with the creation of what we call ketone bodies. Basically what happens is we start tapping into our fat reserves, and we have to break down those fatty acids, and a byproduct of that is ketones. And we're able to use the breakdown of these fatty acids as energy for all of the cells in the body. All right, so the rates of all of these reactions are going to control the amount of heat made by the body, and the amount of heat loss is going to have to equal the amount of heat production in order to maintain homeostasis of body temperature, right? So if you increase your metabolic rate, you're going to probably increase your body temperature, right? And metabolic rate is defined as the overall rate at which metabolic reactions are going to use energy in your body. Metabolic rate has a basal level, so a basal metabolic rate is what happens when you are sitting quietly listening to this lecture, right? And that is going to be able to be affected by multiple different external factors, including things like exercise, or hormones, or nervous system, body temperature. Ingestion of different foods can make you sleepy, or ingestion of different drugs like caffeine can increase your metabolic rate. Your age, so as you hit your mid-30s and onward, your metabolism hits a cliff and then slowly drops off from there. Also your gender, the different climate, your ability to sleep, and malnutrition can also affect all of these different metabolic rates. We have heat loss that happens from our body, and it happens by a lot of different ways: conduction, convection, radiation, evaporation. And we're able to detect our body's temperature based on an area of the hypothalamus called the preoptic area. It's basically the body's thermostat. And these thermoreceptors are there to be able to send information. So there's thermoreceptors all over your body that are going to send signals to different regions, right? So there's a region in the hypothalamus that's the heat-losing center, and there's a region that's the heat-promoting center in the hypothalamus, and that's going to be dependent on what the body needs. And this is going to allow us to have a feedback mechanism that allows us to conserve and increase heat production when we need it. All right, so just like anything else, we have some sort of stimulus that disrupts some sort of controlled condition. So in this case, the disruption of homeostasis is going to be occurring because we have changed the body temperature. So we're starting to get cold. We've decreased body temperature. We have thermoreceptors that are present. Again, no matter what it is that we are trying to maintain in a homeostatic balance, we have receptors to detect it. Otherwise, you would never know if it went awry. So the thermoreceptors in your skin and then also in your hypothalamus are going to go to a control center. They're going to send information through nerve impulses to a control center. That is the preoptic area and also the heat-promoting center in the hypothalamus. And it also is going to be connected there in the anterior pituitary. And the output that it's going to release via nerve impulses and the thyroid-stimulating hormone are going to include a couple of effectors. The first is vasoconstriction. Vasoconstriction is going to allow us to decrease the heat loss through the skin because we're not getting as much blood to the surface of the skin, so we're keeping the blood at the lower levels of the body internally, and hopefully we won't lose as much heat through the skin. We also are going to have an effect of the adrenal medulla, which is going to increase cellular metabolism, because if we increase our overall cellular metabolic rate, we're also increasing heat production from internally. And then we can also tell our skeletal muscles to contract in repeated cycle, and that's called shivering. I'm sure we've all been there before. It's not something that you can control, but what that's going to do is going to physically shake your body to increase your temperature. And also our thyroid gland is going to release some different thyroid hormones, which are going to increase your metabolic rate as well. All of that is going to result in an increase in your body temperature, and it's going to continue in this way until we have retained or returned back to homeostatic conditions in which we have returned to normal body temperature. All right, so as I mentioned previously, if you eat too much and you consume more than you're able to burn off, you're going to end up creating adipose tissue. Right? So we want to make sure that our energy intake is directly is equal to the amount that we send out the door. And that energy intake is directly dependent on the amount of food and the different types of food that we consume. So for example, if you're just going by a pound of vegetables is not going to give as much energy as, say, a pound of fat, right? So depending on what you are bringing into your body, you're going to get a different amount of energy out of that body, or out of that amount of food. The energy expenditure of an individual is based on a couple of things. One is the basal metabolic rate. The other is your physical activity, and something called food-induced thermogenesis, which is basically when you eat, it's going to change your body temperature. There are areas in your brain that control the hunger impulse. Those areas are the paraventricular nucleus in the hypothalamus, and also the arcuate nucleus, again in the hypothalamus. And those are going to be responsible for stimulating your hunger responses, or responding to your hunger responses. The hormone leptin is going to help decrease your body fat mass. Neuropeptide Y is going to stimulate the response for food intake, and melanocortin is going to inhibit your food intake. And we have nutrients that we can take in as well as that are going to be responsible. Basically, they're chemicals that we find in food. So they're small particles that are inside the food that include things like water, carbohydrates, lipids, proteins, minerals, and vitamins. So they're basically what you're going to find inside the animal cell or the plant cell that you're consuming. And then our cells are going to manipulate them and use them for growth and maintenance and repair. So as I mentioned previously, that you are going to be getting different things out of different types of food molecules. So in that way, we have what's called a food pyramid, right? We want you to proportionally intake different amounts up from the different food groups. So we want you to take in 50 to 60% from carbohydrates, and we want them to be complex sugars, complex carbohydrates, not simple sugars. So we want approximately 15% or less than 15% of that to come from simple sugars. We want approximately 30% of that to come from fats or less. And there's two different types of fats, which I haven't really touched on, but we have saturated and unsaturated fats. Now picture that being the difference between bacon grease and olive oil at room temperature. Right? So bacon grease is going to be solid at room temperature. Those are your saturated fats. Those are going to have a different impact on your heart and vasculature than, for example, your healthier fats like olive oil, which is actually not a fat at all. It's an oil, but my point is it's not as saturated. It's unsaturated, and so it's going to be liquid at room temperature. We also want to take in about 12 to 15% from protein. Additionally, we have some other things that we take in that are things like minerals and vitamins. Minerals are anything that's an inorganic element but is involved in specific roles in your body's metabolism. And vitamins are going to be nutrients that are found in really small amounts, but we do need them, so trace amounts, in order to maintain growth and normal metabolism of our body. And a lot of these are things we cannot synthesize and have to be consumed from exogenous sources. So oftentimes this is why we take vitamins, because we might not necessarily get vitamins in our diet and we can't make it from our own body, so we take them as supplements. This is a list of different minerals that are vital to the human body. They include things like calcium, which is actually the most abundant mineral in the body, and you know it as being stored in your bone and in your teeth, but it's also in your blood, and it's all controlled by parathyroid hormone, and we talked about that when we did the endocrine system. But basically, the calcium is going to be absorbed by something called calcitriol, or calcitriol is going to promote that absorption rather, and anything excess is going to get excreted. Again, it's involved in the formation of your bones, but it's also involved in blood clotting responses, which is why it's in your blood supply. It's also involved in your muscle and nerve activity, endocytosis and exocytosis, which is whereby things are coming into and out of the cells, the ability of cells to move around. It also is involved during mitosis, so the way that the chromosomes move during mitosis and meiosis, and metabolism of glycogen. So as you can see, these minerals, even though they are found in small amounts, are going to be super important in these major metabolic pathways. Phosphorus is also found in the bones and the teeth, but we see phosphates a lot in the buffer system of the blood, right? You may remember when we talked about fluid balance. Also responsible for muscle contraction, found in many enzymes, and is a major component of DNA and RNA. It's also going to be found in all of your, I'm surprised I didn't mention that, it's found in your plasma membranes as well. Alright, so potassium is going to be really important as well. It's a cation. It's depicted as K, and it's going to be present in a lot of different foods, right? Bananas, for example. So lots of different fruits. And it's needed for conduction of action potentials in your neurons. We also have sulfur that is found in many different proteins. Anytime you see something that has a disulfide bond or has cysteine in it, cysteine is going to have a sulfur in it, so it's an amino acid that has a sulfur inside of it. It's also involved in electron carriers, so a lot of the electron-transport chain relies on sulfur, and it's found in several different vitamins as well, like biotin. You can find it in sets of meat, basically, meat and animal products, but also in some things like beans, so some vegetables. It's needed for hormones and vitamins and proteins and all sorts of things, and needed for ATP production, again, in the electron transport chain. Sodium we've talked about extensively throughout this semester, so you guys should off the top of your head be able to give me 4 or 5 reasons why sodium is important in the body, but it's found in the extracellular fluids mainly, and it's also going to be found a little bit in bones, and it's involved in concentration of fluids and perspiration, etc. And it's going to help with water distribution and osmosis involved in the bicarbonate system as well, and also in nerves and muscle action. So it's going to be very important. And chloride is going to be involved in a lot of the same things. It's going to be the major anion in your extracellular fluids. So sodium and chloride often go hand in hand. And again, they're found together, sodium and chloride. And they're going to play a role in the acid-base balance of food because of the way that the hydrogens are going to be able to bind to it, and also the formation of hydrochloric acid in the stomach. Some other minerals are going to include magnesium and iron. Magnesium is really important for muscle tissue, nervous tissue, helps create bones. It's also a coenzyme, so you see magnesium a lot when we talk about coenzymes and different metabolic pathways. Iron, super important because it's found in hemoglobin, so it's involved in oxygen transport. It reversibly binds oxygen, so it can give it up or bind to it, and it also can bind carbon dioxide, and it's also involved in those cytocrines that again are involved in that electron transport chain. So as you can see, minerals are going to be very important in a lot of metabolic pathways as well. Some other minerals include iodide, which is involved in regulating metabolism, manganese, which is needed to synthesize hemoglobin, copper, which is also required for synthesis of hemoglobin, cobalt, which is required for erythropoiesis, which is the creation of red blood cells, zinc, which is important in carbon dioxide metabolism, fluoride you may have heard of, they add it to the water. It's going to help with tooth structure and inhibit tooth decay. Selenium, we actually have a problem with fluoride right now because most people are drinking bottled water, and so they're not getting enough fluoride in there, and so they are having softer teeth, but I digress. Selenium is really important for sleeping, right? Does it say that up here? It doesn't, but it's important for thyroid hormones, sperm motility of the immune system. It also functions as an antioxidant as well, and it's also been known to help play a role in preventing certain problems like birth defects, miscarriage, and coronary artery disease. Something called chromium is going to be responsible for regulating insulin in lipid and carbohydrate metabolism. Alright, so let's switch from minerals for a second over to vitamins. There's a ton of different vitamins that are in the body, and they have different functions. Some of them are going to be fat soluble, and some of them are going to be water soluble. So our fat soluble vitamins are going to include vitamin A, vitamin D, vitamin E, and vitamin K. Vitamin A is going to be required as an antioxidant. It's also really important for vision. So I may have mentioned previously the GMOs and the golden rice. It's going to be responsible for creating beta carotene, essentially, which is going to help with the photoreceptors of the retina. So it's going to help with distinction of color versus black and white vision. And it's also going to aid in the growth of bones and teeth. Vitamin D you've all heard of, right? Drink your milk. So you can get it from milk, but you can also get it from by going outside and converting it using sunlight. It's going to convert precursors into vitamin D. And it's important for absorption of calcium and phosphorus. So that's why it's really important that it's in the milk, because it's going to help us absorb the calcium inside the milk in order to get it into our system. It also is going to work in concert with parathyroid hormone. Again, we talked about that when we did the endocrine system. That's going to help us maintain calcium homeostasis as well. And if we have issues with that, we end up with rickets, which you may have heard of before. That's what we call it in children. It's called osteomalacia. In adults, it basically what happens is your bones get very, very weak. Oh, sorry guys, let me go back. Vitamin E you've probably heard of in terms of scarification. So you will put this on top of post-surgical scars or tattoos because it's going to help with healing. And it's also involved in the formation of DNA and RNA and red blood cells. Vitamin K is first produced in your intestines, and it's going to be a coenzyme that is involved in clotting factors, and it's going to associate with prothrombin to help with clotting factors. So if we don't have vitamin K, we can end up with a reduced clotting time or a delayed clotting time, and that might lead to excessive bleeding. Let's talk about the water-soluble vitamins for a second. So water-soluble vitamins include B1 or thiamine, B2, riboflavin, niacin, and B6. And you've probably heard of a lot of these, maybe just kind of as a cursory, maybe not in depth. The water-soluble vitamins are going to be the ones that can be carried by the bodily fluid and are not going to be stored in the body. And these are going to be the ones that, so if you take too much of the fat-soluble vitamins and it doesn't end up excreted, you can end up having toxicity occur. It's very difficult / borderline impossible to have toxicity of a water-soluble vitamin because the excess intake is going to be eliminated easily in the urine. Okay, so B1 is a enzyme, and it's involved in carbohydrate metabolism. B2 is also a coenzyme component that's involved in carbohydrate and also protein metabolism. It's found in the eye, the alimentary system, so skin, right, mucosa, the intestines, and the blood. Niacin or nicotinamide you've heard of because that's found in NAD. It is the N in NAD and NADP and NADH. These are all different types of coenzymes that are involved in redox reactions that we've gone over quite extensively at the beginning of this reaction. It's also involved in lipid metabolism. Vitamin B6 is also a coenzyme involved in amino acid metabolism, and it's going to help you create circulating antibodies, and it's also involved in the metabolism of triglycerides. Some other water-soluble vitamins include vitamin B12. This is going to be a coenzyme that we need to be able to create red blood cells, also to form the specific amino acid methionine, and also involved in the entrance of particular amino acids into the Krebs cycle. Remember I said that we can use alternate forms if we don't have enough glucose to be able to get our energy, to meet our energy needs, and so it's going to be responsible for helping to pull those amino acids into the Krebs cycle if needed. Additionally, it helps make acetylcholine by creating choline, which is a precursor. Pantothenic acid is a water-soluble vitamin as well. It's going to be a part of coenzyme A. We just talked about why coenzyme A is important because it brings, it basically transfers the acetyl group onto pyruvic acid to help bring it into the Krebs cycle. We've also talked about folic acid. Folic acid is responsible for the production of red and white blood cells. What you may have heard of with that is that we talked about it during our development lecture, because if you have a folic acid or folate deficient as a mother, you can end up with issues with neural tube defects in babies, such as spina bifida. Biotin is going to be found in your GI tract. It's also a coenzyme. It's going to help convert pyruvic acid to oxaloacetic acid, which is going to be part of the Krebs cycle, obviously, and it's also going to help synthesize the fatty acids and purines in our body. Last but not least, we have vitamin C. You've probably heard of this before. It's ascorbic acid. Ascorbic acid is going to be responsible for helping promote protein synthesis, and it's a coenzyme, and it has often been used in treatments for people who have been poisoned because it can help offset the harm of the poison by rendering it harmless for a little while until the poison is able to be metabolized and excreted in the body. Okay guys, that brings us to the end of our metabolism lecture. I really appreciate you sticking with it this whole time. I hope you have a fantastic day. Take care.